All right, welcome to our Introduction to Cells lab. Here are materials for this week: newspaper, scissors, compound light microscope, toothpick, methyl or methylene blue, glass slide and cover slip, those should be blank, and an onion. Okay, so starting with part 1, we're going to take a look at the letter E from a newspaper under the microscope. So cut out the smallest letter E that you can possibly find on your newspaper. And a magazine will not work because the paper is too thick and the light from the microscope won't pass through it. So cut out your E. It should be super tiny. And you're just going to want to put it on your slide. Just like that. And then we're going to put it under the microscope. And you're going to use low power, which on my microscope is 4 times magnification, that lens. And then again, you're going to magnify it by the low power times the eyepiece, which is an additional 10 times. That'll be 40 times on low power. Okay, so this is what it looks like under my microscope. I'm starting with the stage all the way at the bottom and using the large knob, the coarse adjustment, to bring the stage up slowly until the newspaper comes into focus. And then I'll use the smaller knob, the fine focus, to make it nice and crisp once it's as close as we can get it on the coarse adjustment. And you'll notice that you can see the newsprint E, and it is upside down. And that's because the lenses on the microscope flip objects over. Okay, on to part 2. We're going to look at human cheek cells. So you're going to take a toothpick and rub it on the inside of your cheek until it's wet and covered with some of those dead skin cells on the inside of your cheek. And you're going to rub it on the slide. Should be a little bit of a film on it. And then you're going to put your cover slip on, and we will create a wet mount, a stained wet mount of these cheek cells, because they are clear otherwise and pretty hard to see. You can certainly see them if you don't have stain, but it's going to be a little bit harder. So we use the methyl blue or methylene blue, and you're just going to put 1 drop on one side of the cover slip, and you're going to tilt your slide, and with a paper towel you are going to draw that stain through the specimen, and then the paper towel will absorb the extra stain. If you just plop a drop of stain on top of your cheek cell specimen, it will flood the slide and you really won't be able to see anything very well. Things will be moving around because it's just too sloshy and wet. So get as much of that stain on the paper towel as you can, and then you're going to put it under the microscope. And we always start on low power, but this time we'll bring it all the way up to high power. Okay, so we are starting on low power, and again starting with the stage all the way down. And then as you're looking through the eyepiece, you're focusing and bringing that stage up until something comes into focus. And here you'll see our cheek cells, which are stained slightly blue. Yours might be a little bit darker depending on which kind of stain you used and what the concentration is. But so we're going to focus even higher on